My name is Brett Sansbury, and I will be giving a brief overview of our work enhancing COVID-19 genomic surveillance using several CRISPR-based molecular techniques. Particularly, I will be talking about our approach to investigating the robustness of two CRISPR-based tests for COVID-19 detection. These are STOPCovid and Detector. We first validated that these tests using synthetic COVID-19 RNA and assessed the lower limits of detection for both methods. Here you can see a comparison of both of these testing methods with a notable difference between them being that each uses a unique Cas12 protein. Cas12a is used for detector and Cas12b for STOPCovid. As compared to standard PCR protocols, each test is essentially faster and more capable of being run without sophisticated training or equipment. As we have seen in recent weeks, mutations in the COVID-19 viral genome have essentially become a focus of concern for scientists in both the potential infectivity and for their potential for evading our current vaccination strategies. So utilizing these simple tests, we were able to test multiple strains of COVID-19 RNA from variants ranging from unique geographic locations. Some of the available synthetic COVID-19 strain variants that we utilized included those from Wuhan, China, and the US, Texas, and California strains. Here we are showing an example of a limit of detection test between STOPCovid and Detector done with the Wuhan, China strain. As you can see on the left, detection begins as low as 50 viral copies for the STOPCovid, and on the right, Detector testing shows very little detection at 500 viral copies and drops to undetectable at 100. But importantly, here we note that as the lower limit of detection can be seen with tests done with the STOPCovid method, the duplicated tests at these lower viral copy levels are not always accurate. Lastly, here we show an example of a second strain we tested with both CRISPR-based methods, the US Texas strain. On the left, similarly, the STOPCovid testing has still detected samples with 50 viral copies, although again, the band intensity varies at these lowest limits. And on the right, the detector test begins to actually taper off at around 1,000 viral copies for two separate guide RNA sites that we found to assess. So in wrapping up, we have shown here a validation of the STOPCovid and Detector CRISPR-based COVID-19 detection methods. We were also able to assess the lower limit of detection for each of these tests separately, and additionally investigate the sensitivity of each test to different variant strains of COVID-19.